We see that it's important to take both the biological sex but also the socio-cultural gender aspects into account when working with research and innovation, so that solutions, targets, and are relevant for a larger group in society.
Incorporation of questions concerning the potential of sex-related differences in drug response early on in drug discovery and development is not just a matter of good practice, but is also very much needed for the personalized treatment in the future. In recent studies, we have been also looking in oxycodone. This is opioid analgesic, and interestingly, in healthy conditions, we could not see any differences in pharmacokinetics between male and female rats. However, when we have induced a pathological condition, systemic inflammation, by introducing bacterial endotoxin, we have seen dramatic differences, and in this situation, female rats had slower elimination of oxycodone, leading to higher concentrations, which is rather similar to the clinical observation situation. So this is highlighting, first of all, similarities between preclinical and clinical observations, and also the need of studies in pathological conditions as well, not only healthy conditions.
We can definitely say, yes, there is a difference in immunological responses between men and women. Women mount an improved IgG response, antibody response to vaccines. And we do also know that women tend to have more frequencies of autoimmune diseases than men, also, telling us that there are differences between the immune responses. And it's also because of sex-linked genes coupled to the X chromosome that we see this.
Historically, women have been excluded from clinical trials due to multiple health-related concerns. However, this trend has been also reflected in preclinical studies. For example, traditionally in a field of pharmacokinetics, many studies have been performed only in healthy young male rodents. We have recently observed dramatic changes in related to this question, and this is governed by various aspects. For example, funding bodies, they require inclusion of both sexes in the studies and justification of the selection of the sexes if it is only based on one sex.
At Vinnova, we have a mandatory requirement for all our applicants that they have to provide an analysis of sex and gender aspects related to their project, and to describe if it's relevant, and then describe, if it is, how will they take that into account in their research and innovation project and performance. There are differences, both biological differences and socio-cultural differences between women and men, in humans, in animals, also in stem cells. Not always, but there are. And we have seen that it decreases the risk of coming up with unfit solutions that are wrong, solutions that might create more side effects. So this is the most important reason for requiring applicants to take into account, to look at their project proposals and to look at their research and innovation through a sex and gender lens, to see if there are aspects that are important to take into account.
Although there is a huge number of publications that they are addressing this question systematically, we still see a big gap that is related to reporting of disaggregated sex-based studies results. And there is definitely a need of more systematic improvement of this question on all the levels.
And when we analyze our projects, at the end of the project, we see that project teams that describe that they have integrated this perspective, they also describe a deeper understanding of the problem that they focus on. And they come up with more relevant solutions for a larger group of people, target groups, end users. So we see that it's an important aspect of the quality of the proposal.
We try to, when we use models in vivo, to think about the gender dimension. So indeed we have both male and female mice in animal experiments. And we do this more and more from a perspective that more and more evidence points to this type of subtle differences. It's not so easy for us to maybe take this into account in every detail, because first of all we have to establish efficacy of a drug, and then subsequent to that maybe we can also further increase our understanding of the mechanism to that drug, and then we dig into the sex differences as well.
There are multiple examples where there is variability in the drug response between women and men. The differences could be explained by various factors and in part related to pharmacokinetics, which influences drug exposure over time, and also pharmacodynamics, which influences drug safety and efficacy. In our work and in my own research, we study pharmacokinetics and pharmacodynamics of drugs in different aspects, and we have several examples where we have seen differences between sexes. In one of the earlier studies, I've been looking in general anesthetic propofol, which is widely used also in Sweden. And there have been multiple reports showing that women require higher doses to induce anesthesia, and they also recover faster from anesthesia. So in an observational study, we have identified that women metabolize propofol faster, which could in part explain this variability between sexes.